Hello. So today I'm going to be talking about the cell biology portion of my e-report. So first I'm going to talk about cell membrane structures with different classes of membrane proteins and energy requirements. And you can see on the right there's a picture of the cell membrane, and you can see within it there are proteins. These are membrane proteins, and this is overall the structure of the cell membrane. So the cell membrane is a part of the cell that separates the outside world from the inside of the cell. So the cell membrane is known as the protector of the cell because it protects the ordered biochemistry life in the cell from the chaos and disorder of the non-living universe outside. So the cell membrane is a mechanical marvel, and it's just because it shapes, it stretches, it flexes, it grows, it deforms without tearing, and it even has the magic ability to be self-sealing. But let's give the cell membranes a little more credit than that though, because they are even the gatekeepers of the cell, allowing only certain molecules to enter and leave. Therefore, since the cell membrane does all of that, it has to be a portal and yet at the same time a barrier. So lipids are the barriers and proteins are the portals. But let's back up a little and talk more about the structure of the cell membrane. So the cell membrane is known as a lipid bilayer because it is made up of two opposed layers of lipids. So this bilayer is what makes up the barrier between the inside of the cell and the outside of the cell by blocking the diffusion of most hydrophilic molecules because lipids are classified as being water insoluble or hydrophobic. But the lipids that make up the cell membrane are different in that they have a hydrophilic head seen on the right and the top, containing charged groups, and a hydrophobic tail on the hydrocarbons, which are in the center on the right. These membrane lipids or phospholipids are amphipathic because they have both the hydrophobic and hydrophilic parts that drives this membrane's formation. So, when you place the cell in water, the cell membrane phospholipid tails, because they're hydrophobic, are going to run away from water, while the heads are going to face the watery environment, both inside and outside the cell. And this forms a hollow ball known as the liposome, seen right here. And once we get bigger and full of life, and we have proteins and nutrients and DNA and RNA and all these wonderful organelles, it's the lipid bilayer. Because this lipid bilayer can form a ball with no free edges, it's considered self-sealing and can conserve energy. Therefore, no energy cost to create the shape of the cell or seal any damages. What amazing. So, since there are hydrophobic regions within the phospholipid of the membrane, they're never fully happy, and this shield from water is exactly why nothing pops out of the membrane. Even though they are stuck, these membrane lipids and proteins can float forwards, backwards, and side to side, which is known as lateral diffusion. The ability for the cell membrane lipids to laterally diffuse is why the membrane is a two-dimensional fluid. So, these membrane components can flip upside down once a month in a process known as transverse diffusion. But the accidental flip is fixed by these wonderful flipases. And basically what they do, they're embedded in the membrane to restore the correct position of the membrane. So another great thing is that this membrane is a two-dimensional fluid, and fluidity is used a lot with the membrane. And it's thought about in a sense of how loose or stiff the membrane is. So think about it. More fluid membranes are less dense and easy to float in. So, these fluid membranes have less packing, shorter tails, and more double bonded tails. Less fluid membranes are more dense and harder to get around. They're very viscous. These less fluid membranes have more packing, longer tails, and less double bonded tails, which would give you more room. Since we cannot manipulate the hydrocarbon tails or length of double bond numbers, we just regulate the fluidity with cholesterol. These phospholipids are pretty much structural in the fact that they create a permeable membrane. But the portal, the activity, and the function of the cell membrane is carried out by these wonderful membrane proteins. And the membrane protein's main function is just to transport molecules inside or out of the cell that would not be able to usually diffuse across the membrane. And there are different types of membrane proteins. So a broader terminology of membrane proteins are peripheral proteins and integral proteins. So peripheral proteins are proteins that sit on the surface of the cell membrane or are not embedded at all in the membrane. So a good example of these proteins are actually hormones. They sit on the cell to relay some type of message to these cells. These peripheral proteins are not involved in diffusion of molecules, so they don't require energy. There are also integral proteins, which are proteins that are embedded inside of the cell membrane. So there's two main types of integral proteins. There are channel proteins, which is almost like a doorway. It's seen on the left here. And the fact that it just allows big charged molecules to flow in and out of the cell following the concentration gradient. So we're going from low to high concentration. And this is done in facilitated diffusion and passive transport. So because we're doing passive transport, these channel proteins do not require energy. Mostly because these molecules are following the concentration gradient. So there's also transport membranes, and these are also seen in the picture as carrier proteins, and that's considered a pump because it's actively transporting and facilitating the diffusion of molecules movement in and out of the cell, but against the concentration gradient. So we're going from a low to high concentration, so that's not what the cell wants. So it's actively transporting. We're still facilitating diffusion through these carrier proteins or these transmembrane proteins, and it's requiring energy. So next I'd like to talk about the formation, the docking, and the fusion of clathrin-coated vesicles. So first, transport through the entire endomembrane system is just carried out by a continual process of budding and fusing and involving transport vesicles. And these vesicles move in two general directions, from the ER, or the endoplasmic reticulum, to the Golgi, and to other destinations in the cell, or from the membrane to the lysosomes and further inward. So this vesicle movement provides routes of communications within and throughout the cell. So this vesicle transport between organelles is highly organized and choreographed mechanism, and this is for these hydrophilic proteins and membrane components to move around the cell. So challenges involving vesicles are when it buds off an organelle, it has to only take the intended protein, and it needs to be specifically routed to the proper destination as well. So these challenges are actually fixed by a protein coat that covers most vesicles, known as the coated vesicles. So this coat actually helps form the bud. So you see right here the vesicle formation. This green is the clathrin coat, which is a protein coat, which is what I'm talking about. So this coat actually helps form the bud, hold it in shape, target the vesicle, and ensure it's in the right contents before fusing with its target. And then the vesicle sheds this protein coat. It has everything it needs. It knows where it's got to go. Now when we get there, we got to get rid of it. So the formation. So at the cell membrane, each vesicle starts off as a clathrin-coated pit, which is like a basket structure with the clathrin protein mesh at the base of this pit, which starts to shape the vesicle, which you could start to see right here. We have this in the beginning, we have this start of it. So there's also a second class of vesicle-coated proteins known as adaptins. And these adaptins are seen in that light green, and that secures at the clathrin cage to the vesicle membranes, and it is needed for cargo selection, which is loaded into the vesicle. So that's exactly like what we're putting into the vesicle to be transported. So these proteins that have their destiny set out for other targets, they know where they have to go. They actually have transport signals on them, and these transport signals bind to the cargo receptor. So this cargo receptor is shown in blue, and the cargo molecules that are going on it are in red. And so each type of adaptin binds to a specific set of cargo receptors and captures the cargo molecules by trapping them below the neck of the budding vesicle. So then a small protein shown in the second portion of this, known as dynamin, will form a ring around the neck of the vesicle, pinch it off from the rest of the membrane, and this is how the vesicle is formed. So that would be shown in the third portion of this picture. So then we uncoat, as you could see. So for docking, which is next, once the vesicle is formed and it's reached its target organelle, it must recognize that the membrane, and it has to dock to it. So luckily, there are specialized proteins called Rab proteins, which is seen in yellow, on the membrane of the vesicle specifically, and it gives its insight on its origin and the cargo it's carrying to know what membrane it has to dock with. So these Rab proteins are essential because the vesicle membrane proteins will recognize and bind to tethering proteins that are present on the destination membrane, which is seen in green here. And it's on the target membrane, and that's what the Rab recognizes. So these tethering proteins will only bind to Rab proteins that come from places that should dump off their cargo at this site. So tethering proteins have a say too. They're not going to bind with Rab if this is not where they need to go. So this interaction between Rab proteins and tethering proteins is essential for docking, and it assures that only the vesicle with appropriate original organelles fuse with the correct target membrane. So there are also snare proteins, which are also known as V snares and T snares. V snares are vesicle snares on the vesicle, and transport snares are at the target membrane. And that's for additional recognition, and it stabilizes the docking process, and it even adds a second layer of anchoring, so that vesicle is not going anywhere. So there's also fusion here. So the membrane must fuse, connecting the vesicles to the target and release the vesicle content to the appropriate destination. So once fusion occurs, the vesicle membrane will become part of the target membrane. They are one thing now. It is important to note that sometimes fusion is delayed and allow the vesicle just to be docked, but it needs to wait for a cellular signal for fusion to be initiated. And this is widely true for cellular secretion. So for membrane fusion to occur, we have to get the vesicle and target membranes so extremely close to one another, even further than the closeness that was caused in the docking previously. But how do we get so close that fusion can even happen if we need them adjacent to one another? Well, that's what the V snares and the T snares do. As you can see, they're starting to twist around one another, and it brings this vesicle in, and that vesicle will then be fused into the membrane. So now I'm going to talk about the formation and the structure of all three types of cytoskeletal fibers. So this is a picture of the cytoskeleton, and the cytoskeleton is a meshwork of protein filaments, and that permeates all aspects of the cell. So it's an amazing structure. It's highly dynamic in that it changes shape in response to signals. It mediates the location of interior components. It mediates the interactions with the environment through reacting and protruding structures, and it even mediates coordinated movement. But even more amazingly, it's a major component for cellular transportation, and it pulls chromosomes apart and splits the cell. Imagine if mitosis couldn't happen. Ooh, man, I don't want to think about it. Cellular mobility and motility would completely be impossible without the cytoskeleton. The cytoskeleton is built by 3 different protein filaments and are all specialized in different ways to perform a specific role in the cytoskeleton. So the first one I'm going to be talking about, the first protein filament, is intermediate filaments. So intermediate filaments, or IFs. IFs are the strongest, toughest, and most durable filament in the cytoskeleton. Therefore, their specialty is tensile strength within the cytoskeleton. Its main function is to help the cell withstand the mechanical stresses, such as stretching, pulling, squeezing. And without these IFs, the cells might not be able to survive these stresses of the mechanical activities. So these IFs are found throughout the cytoplasm, forming a network surrounding and anchoring at the nucleus and extends out of the cell membrane. So these IFs are even found inside the nucleus, where it is the main component of the nuclear lamina, or the cortex of the nucleus, however you want to think about it, to strengthen the nuclear envelope. So, how are they formed? These IFs are made like ropes. 2 IF subunits twist together to form a dimer, and then these dimers are put together in a staggered form to make a tetramer. And once these tetramers bundle together, they form an IF. And the heads of these IF proteins are globular, and they allow them to interact with many things. So, there are 4 types of IFs: keratin, vimetin, neurofilaments, and nuclear lamins. Now, all of the first 3, keratin, vimetin, and neurofilaments, are formed just like the rope. But the nuclear lamin is a little different. It forms a 2-dimensional mesh. It is a wicker basket, and it's perpendicular. And since they are a major component in the nuclear envelope, they are formed like this specifically because they need to disassemble and reform with each round of cell division. And it's controlled by phosphorylation. So the second protein filament that I'll talk about that makes up this cytoskeleton is the microtubules. So these microtubules, or MTs, play a major role in the organization of the interior of all eukaryotic cells, and they anchor and localize organelles to allow walking proteins to move cargo around the cell, transporting the vesicle, just like I talked about in the last topic. We're moving those vesicles. They can walk. They can walk on microtubules. So microtubules are long, stiff, hollow tubes of proteins, and they can rapidly disassemble at one location and assemble elsewhere. So these microtubules have a centrosome, which is shown on the right up at the top. And this is their point of origin and allows them to grow out of it and near the center of the cell. So these microtubules that extend out of the centrosome to the interior face of the cell membrane is what creates the tracks that allow proteins to walk on them for transportation of vesicles. How great is that? These microtubules are made up of a subunit protein called tubulin, which are dimers of two globular proteins, an alpha and beta subunit, which you can see on the left picture on A portion, or you can somewhat see it in the picture on the right underneath, where the GTP-B tubulin, or beta. So these dimers stack together, interacting through non-covalent forces with the long chain of globular spheres alternating between the two subunits, so alpha beta, to form part of the wall of the microtubule. So it takes 13 of these protofilaments, which can be seen right on A next to the dimers, to interact side by side to make this single hollow microtubule. Since these protofilaments are alternating between alpha and beta subunits, they have a beginning and an end, which is great because it gives microtubules polarity. So tubulin can be either added to either end, but it prefers to grow from the growing end or the positive end. So this polarity, which is the positive and negative end of microtubules, is crucial for allowing the proteins that walk on them the directions to ship their cargo. So these microtubules form rapidly when dimers are added to the pre-existing microtubule fibers, but cannot form readily from scratch. But here comes the centrosome for the rescue, because it serves as more than just the point of origin, but also a tubulin organizer, and it coats the tubulin to form the initial microtubule tube. So the centrosome contains hundreds of rings that are made up of this third type of tubulin, and these rings serve as the nucleation sites, and that's the beginning of the microtubule formation. So the negative end of the new microtubule is embedded into the centrosome, and the positive end points outwards towards the cell periphery, and it just grows and grows and grows. But, the entire microtubule can just disintegrate without even warning. Tubulin dimers bound to GTP have a high affinity for joining other dimers, but this dimer will eventually cleave the GTP to GDP, resulting in the loss of affinity. And it will stay there as long as the overall microtubule is kept stable by GTP cap, or anchoring. Once microtubules fall apart, the dimers become free, release GTP, and take on a new GTP molecule that has a high affinity for joining a microtubule. They want to join. This notion of tubulin dimers being inherently unstable and a microtubule's overall default state to the falling apart is referred to as dynamic instability. How great is that? So we can stop these microtubules from falling apart by anchoring the positive end to something. The cell has no way to target a microtubule to a specific location, but the centrosome sends out new microtubules. Therefore, the ones that catch a good target, great, I'm stable. And the ones that miss a good target, retract to the centrosome, and they are depolymerized. So you can see on the picture on the left, we have polymerization to form it, and then we have depolymerization to take it apart. So this allows the cell to be highly organized in interconnecting systems of protein fibers. And the polarity of the cell is reflected on the polarity of the microtubules, which helps guide this vesicle transport in positions and segregates organelles correctly. So the third type of protein filament that I'll be talking about that makes up this cytoskeleton is actin. So actin is the smallest filament in all your eukaryotic cells, and it's essential for almost all of the cellular movement. Without actin, the cells would not be able to crawl along surfaces, perform phagocytosis, or divide into two after mitosis. These actin filaments are inherently unstable, like microtubules, but if bound by other proteins, actin will be stabilized. Each filament of actin is twisted chain of identical globular actin subunits, and they also have polarity and that points in the same direction, much like these microtubules, but these actin filaments are thinner, shorter, more flexible, and they're not hollow. Actin filaments are typically bundled together in a cell and not often found on their own. So without being associated with these accessory proteins, the actin filaments will disassemble easily from both ends. Each actin monomer is bound to ATP, and this is what you can see in the left schematic. And once incorporated with this filament, ATP is cleaved to ADP, reducing affinity and promoting depolymerization, which is seen on that minus N on the right. So if accessory proteins are bound to actin filaments, the accessory proteins modify these actin filaments and cause these actin to be more stable. So actin makes about 5% of the total proteins, half in filaments, half in free monomers, and the high concentration of free monomers is intentional. And this is to allow small proteins in the cytosol to bind to these actin monomers to regulate this polymerization by stopping free actin to add on to growing filaments. Imagine if we didn't have this free monomers, we would just keep polymerizing and polymerizing and polymerizing. We would never have any type of regulation. So when new actin filaments are needed, proteins release their monomers, actin monomers bound by other proteins promote assembly, and these new filaments are made. Most accessory proteins are already polymerized and have multiple functions, but actin bundling proteins hold actin filaments together in bundles. So other proteins hold actin into a meshwork of the cell cortex. Filament serving proteins cut actin into smaller pieces and convert it to a more fluid state. So remember, we're still in portions of like the cell membrane and the cell, and we want that fluidity. And actin can form tracks when bound to motor proteins. So actin is a protein that is most concentrated underneath the cell membrane. And why is that? It's providing support, and these actin filaments link in a meshwork by accessory proteins to provide mechanical strength, dictate shape, and explains the dynamics of this beautiful cell. So next thing I'm going to talk about is cell crawling via actin, of course. So many cells move by crawling towards sensing environmental signals by using actin in a 3-step process. So the cell pushes out protrusions at its leading edge. These protrusions adhere to the extracellular surface where the cell is crawling, and the rest of the cell flows, quote unquote, forward to these anchoring points, filling in the cell at that advancing front. And these 3 processes involve actin differently. So we're using actin differently in each one of these steps. So the first step is driven by new actin polymerization, where the lampodia, which is a dense meshwork of actin, is pushed out, and the actin is oriented where the positive end is close to the plasma membrane, and this is seen on the top portion of this figure. The smaller protrusions, the filopodia, reach upward and outward with their positive ends on loose bundles of actin. So this can be seen by the little red in the second portion of this. It's like reaching out from the lampodia. So this actin polymerization of these structures cause the cell membranes to push out. So the crawling cell, actually, will constantly send out these structures all over the cell surface, sense the chemoattractant, and anchor to the crawlable surface. And this is a reversible process too. But you can kind of see these focal contacts that they have. That's basically being sensed by these chemoattractants that are there for it to anchor to this crawlable surface. So the anchoring is actually done by membrane proteins in the cell membrane known as integrins. So these integrins adhere to molecules that are present in the cell's ECM, and the integrins are directly anchored to actin filaments in the intracellular side of the membrane. Therefore, integrins bridge actins to the surface that they're being crawled on in this process. So the cell will then pull itself along the surface, involving the contractile forces between actin and myosin. So myosin and accessory proteins regulate this actin behavior. We always have to regulate everything, because if you don't regulate, we lead to things like cancer. But everything in the cell is responding to extracellular cues so that the cell rearranges its cytoskeleton in response to these cues. These cues are signal molecules that are mediated by membrane receptors. All the cues that affect actin will accumulate on Rho, a GTP binding protein. Activated Rho will trigger this actin polymerization and bundling for formation of the filopodia, the lampopodia formation, the actin and myosin interactions to promote cell crawling. So without Rho, we can't trigger this actin polymerization, get this formation of the filopodia, the lampopodia, or even have actin and myosin interact, which is bad. We would never have cell crawling or movement within the cell. The integration of signals from extracellular and intracellular environment comes together to affect Rho. If Rho is activated, the cell will move. All cellular movement requires the manipulation of myosin-actin interactions. So, myosin protein families are consisted of two main characteristics. Keep it in mind. They have to bind to actin, and they have to be a motor protein. So myosin binds and cleaves ATP, and this energy allows the movement along actin filaments from the negative to positive end. So there's myosin 2, which is a major myosin protein type in muscles, and myosin 1 that is found in all cell types and allows morphological changes. Myosin 1 has a head domain that binds to actin and bind and cleave ATP, and a tail domain that determines what myosin will move. So myosin has two rigid lever arms that is connected by a joint. So when ATP is hydrolyzed, the lever arm will protrude out of the head domain of myosin, and after the hydrolyzation, the lever arm will rotate, form a bend, and this binding of ATP decreases, and this myosin affinity for actin, and allows the bind-pull-release mechanism of motion. So this is seen right here. So this is what I was just speaking about with the bind-pull-release motion. And this is more specific for muscle contraction, but it's important to mention as well. So next I'll be talking about G protein coupled receptors and including CAMP and calcium as second messengers. So seen on the left is a G protein coupled receptor, and they are the most widely used transmembrane receptors, and they're used to carry the signal into the cell by a series of modified proteins. So these G protein coupled receptors are the largest family of cell surface receptor proteins and are members of the 7 transmembrane receptor proteins. And they play a major role in every aspect of cell communication, and the signal molecules that interact with and bind to these G protein coupled receptors are varied just like the systems they belong to. So they're transmembrane proteins found at the cell surface that were synthesized at the rough ER, threaded through the ER by a start and stop transfer sequence, shipped to the Golgi, processed and shipped to the cell membrane by transport vesicles. Everything's tying together. When a signal molecule binds to its receptor, the receptor changes its shape and activates a G protein that is associated with the receptor at the cytosol face of the membrane. There are numerous different types of G proteins, but they all bind to GTP and GDB. every G protein has an alpha, beta, and gamma subunit. So they are a 3 subunit molecule. Let's see. And when inactive, the alpha subunit will bind to the GDP so that the G protein is dormant. When the receptor has a secondary change in the shape, the G protein decreases its affinity for GDP and increases its affinity for GTP, and then the G protein will let go of that GDP, bind to that GDP, making the protein active. So, once activated, the alpha subunit can alter target molecules, and the remaining beta-gamma complex will do the same independently, initiating the signal cascade. The system is now on, the signals must be turned off, and this is not an issue for G proteins because it has a great system, GTPases activity. And this is what allows it to hydrolyze this GDP to GDP and making it inactive, turning itself off on a timer. These subunits will reassociate, translocate back to the receptor, and repeat the process when a new cell receives a signal. G proteins can activate membrane-bound enzymes or channels. Open ion channels have an effect on controlled enzymes that are used within these G protein coupled receptors. These G proteins affect enzymes at the cell membrane, and they tend to have a more complex and cascading effect. G proteins are indirectly responsible even for the production of intracellular signals. So right here, I want to talk about the 2 most common enzymes to be activated by the G proteins, which are adenylate cyclase and phospholipase C, which the first one is on the left and the second one is on the right, which are activated by different G protein coupled receptors. So adenylate cyclase catalyzes the biochemical reaction to convert ATP to CAMP, which is seen right where adenylate cyclase is, and you see the protein and you see this ATP being transferred to CAMP, which is our second messenger. The receptor, or the G protein, and the adenylate cyclase is stuck at the interior face of the cell membrane, shown in this picture. It's trying to get that message into the cell. So CAMP carries the message into the cell, and this molecule has no information of value. So it just conveys information, so it activates a single protein kinase A, and this kinase A will phosphorylate other target molecules on or off that will achieve this physiological response, or as you've seen in step number 5, effect on cellular function. So there's also on the right the phospholipase C system, and this is where calcium will actually serve as the second messenger and other common G protein coupled receptor systems, and it uses a cell membrane component known as PIP2. So this is shown in the schematic of where 3 is, and what happens is a signal molecule binding to the G protein coupled receptor on the outside of the cell. It activates the G protein, but this time it'll activate phospholipase C. And this phospholipase C splits the PIP2 molecule into IP3, which is right underneath where the number 3 is, and DAG, which is right next to the 3 on the right side. And it serves as a second messenger. So this IP3 will leave the membrane, travel to the cell's interior. But DAG is in the membrane, and it will activate protein kinase C, or PKC, which is seen in step number 4. And this phosphorylation will occur like many target proteins, including these calcium channels that allow this calcium to bind into the cell. So that's what DAG does. So DAG is, technically, to clarify, it's in the membrane, it activates protein kinases, phosphorylates these targets, and it will even phosphorylate specific target proteins, such including these calcium channels, that will allow this calcium to come inside the cell. But IP3 goes straight to the ER, which is shown in number 5. It's going straight to the ER. And why is it doing that? Why is it going there? Well, it's doing that because it's triggering the release of calcium, and the calcium will bind to the PKC and making it extremely more active. So we're having a lot of calcium influx. But calcium also binds to calmodulin, which makes it active. So when calmodulin is active, it activates another kinase, which phosphorylates target proteins that go on to do other things in the cell, or as you see at the bottom, cellular response. Next thing I like to talk about are CDKs and cyclins and how they help the cell progress through the cell cycle. So the primary mode of cyclic regulation of proteins during the cell cycle is phosphorylation, or it being on and active, and dephosphorylation, which is off and inactive. Kinases and phosphatases are the enzymes that are responsible for the phosphorylation and dephosphorylation of the cyclic regulation. These kinases that turn proteins on by phosphorylation are controlled by small signaling proteins known as cyclins. So these cyclins bind and activate kinases, which makes these kinase cyclin dependent kinases because they're like binding together. So you have kinase and cyclin coming together, but kinases are dependent on these cyclins to be active, which kinase cyclin dependent kinases are CDKs. So there are different cyclins that correspond to different phases of the cell cycle. Therefore, the cyclin CDK complex will trigger and phosphorylate different cellular events to prepare the cell for the next cell cycle stage. So this is important because you have specific cyclin CDKs for each phase of the cell cycle. So cells are able to delay the progression to the next phase of the cell cycle if the previous stage is not complete by CDK inhibitor proteins that bind to CDK, blocking the interactions between cyclins and CDKs to pause the cell at the checkpoint. Now that's shown on the right. You could see this inhibition by the CKI, which is just the CDK inhibitor. And I thought that was really extremely cool. It's more complicated, but it really is effective because it's not letting you pass if you didn't basically complete your task or do it properly. So if a signal favors the progression, the inhibitor protein will be removed from the CDK, and if the inhibitor protein is never removed, so if we never remove this inhibitor protein and this is inactive, the cell will either arrest or enter the G0 phase, which means it'll just never divide. So there are DNA damage checkpoints in the G1 and S phase to prevent the cell from starting or completing the S phase if there's DNA damage. So the G1 DNA damage checkpoint is understood better than the S checkpoint. For the G1 DNA damage checkpoint, the DNA damages cause an increase in expression of the activity of a protein P53. This P53 will activate the CDK inhibitor P21, which will bind to the G1S CDK and the S CDK that stops the progression into the S phase. So this allows the cell to just have time. It needs time to repair that damaged DNA. But sadly, if it's taken too long and we can't prepare this damaged DNA, P53 causes the cell to undergo apoptosis. Cell death. What a shame. Without P53 and P21, damaged DNA will be replicated and passed on to the next generation of cell. So normal conditions will allow the cell to pass through these checkpoints. The genome has been replicated, progressed through G2 phase, and it is now ready for mitosis and cytokinesis, which is your M phase. But everything that occurs in the M phase is controlled by a single protein complex known as the mCDK that is formed by the increase of M cyclin levels. So this M cyclin expression begins at the S phase, and it builds through this G2 phase and continually pairs up with its CDK. So the mCDK complex is inactive until it's turned on by CDC25. And this active mCDK will then activate more CDC25s that will indirectly drive its own activation. So this M phase begins with mitosis and prepares the chromosome for segregation. So this chromosome must be copied, and this duplication begins at the start of the S phase, triggered by SCDK and G1S CDK, where the centrosomes stay together until separated to form asters in the beginning of mitosis. So, in prophase, mitotic spindles begin to form by increasing the dynamic instability of microtubules. This increase is partly due to MCDK phosphorylating and inactivating the microtubule binding proteins, making microtubules less stable. So, if you haven't gotten the point yet, these CDKs and cyclins are extremely important to even get things done for this cell cycle. So the microtubules will grow out of one centrosome and touch to grow out of another centrosome at the opposite pole to stabilize the microtubules. And these joined sets of microtubules from opposing centrosomes are the mitotic spindle, and the centrosome is now spindle poles are now anchored to these spindles on either end of the cell. This mitotic spindle are in place. Now we have to attach the sister chromatins to the spindle microtubules so that the chromatins can then pull apart. So I'm adding a little bit of a spin-off of what happens in the cell cycle, but specifically, to get back to it, during anaphase, there's a release of a cohesion linkage by separase. So separase chews up these cohesion proteins to allow the separation of the sister chromatins. And when separase is activated by this anaphase promoting complex, the APC, don't get it confused, anaphase promoting complex, it is targets for the MCDK destruction. So MCDK destruction is the last part of this cell cycle. Once it's destroyed, it's only destroyed when we are beginning the end of mitosis. So, little confusing, but when we're getting near the end of mitosis, we are destructing that MCDK, and then we're done. Our CDKs and cyclins have gotten through all of the phases of the cells. They've regulated, they've made sure everything went done properly, because when it's not done properly and we're not going through these checkpoints, or we're not stopping at these checkpoints if we're damaged, we got cancer. These CDKs and cyclins, their activation and inactivation are extremely regulated and important to be regulated. Because when they're not, I don't even want to know. I'm hoping that never happens to me. So now I'm going to talk about cell junctions. So, there are independent of the ECM pathways that allows these cells to come together and interact. So there are independent methods. There are independent pathways of the ECM, and these allow cells to come together as well. These are cell junctions. And cell junctions in epithelial cells, if you want to be particular. So cell junctions link cells together through physical contact, allow cells to communicate with one another privately, and they allow many mechanical stresses to be shared across multiple cytoskeletons. So I'm going to specifically speak about 5 different cell junction types. So the first one I'll talk about are the tight junctions, which is shown in the middle picture in the top. And these tight junctions seal cells together. And this means that nothing can leak in between them. We have no small molecules going and diffusing between them. It is water sealed tight. So these tight junctions are actually formed by proteins called claudins and occludins. There we go. Sorry. I don't know why I have such a hard time with that word. That are arranged like 2 sides of a zipper on either cell to be joined. And when these proteins zip up, they even define membrane domains. So you see this right here. This is where the red portions in the center, that's causing the seal tightness of tight junctions, and that's done by the claudins and the occludins. So then there are 3 similar cell junctions that hold cells together, known as adherent junctions, desmosomes, and hemidesmosomes. And all 3 of these junctions provide a sheet of epithelial cells with mechanical strength by joining the cytoskeletons together. So the adherens junction and the desmosomes are bound to one epithelial cell to another. And they also, adherins and desmosomes, connect cells through cadherin that are tethered to actin filaments or keratin linking them to the cytoskeleton. So, let's go to the adherens junction. The adherens junction have actin tethered to the cadherin, which is used for cellular movement within a cell when the cell decides it wants to move itself. Desmosomes, on the other hand, which are a little bit down, these desmosomes have intermediate filaments tethered to their cadherins that stop the tissue from tearing and gives them that tensile strength that we want. So then on the left, if you go over here, we have the hemidesmosomes that actually bind epithelial cells to the basal lamina, which is seen right here, the extracellular matrix. The hemidesmosomes have the intermediate filaments shown at the top of the keratin intermediate filaments, and they're attached to these integrins that are attached to the fibronectin that are connected to the collagen that anchor to the ECM. So then on the right, if you go over here, the last cell junction is the gap junction. And this is a private communication system that exists between neighboring cells. And these gap junctions are openings. So this is different from the tight junction because the tight junction is sealed tight. We don't have that. We are closed off, but the gap junctions are actually pretty open. And connexons are what meet these two cells and create a continuous channel across the two cell membranes. And these gap junctions are extremely regulated and can be shut off by cues. So you can see here, these are the connexons. And you can see a wider range over here because you see how it's going through the transmembrane domains. And you can even see it here whether it's closed or open. Is it letting things go in or is it not letting things go in? And this is what I meant by it's regulated by cues, basically. So finally, I mentioned it throughout, when things aren't regulated properly, things are dividing we got cancer. So let's talk about cancer as a sociopathic cellular condition, because that's exactly what these cancer cells act like. So normal cells always work for one major cause. They benefit and help the organism survive. This means that if a normal cell has an issue functioning properly, it will result in apoptosis for the greater good of the organism. We want this organism to survive. That's what these normal cells are doing. So if I'm a bad cell, I'm going to eliminate myself. Well, guess what? Cancer cells are not like this at all. Instead, they are all about themselves, completely selfish. They want to live and don't care if the organism falls ill or dies. It's not their problem. They're all about themselves. They want to be the best. They want to survive. So actually, natural selection and evolution even favors these cancer cells more because they survive better. They overtake and they outcompete the normal healthy cells. These cancer cells are really super cells because they can rapidly divide, they are invasive, they attract a blood supply, they have the ability to grade the ECM, and even have the ability to enter the blood or lymph. So, cancer cells have 6 main sociopathic or lack of empathy cellular conditions. So I'd like to talk about them. So the first one is that they no longer require signals from other cells to thrive. Cancer cells don't use their receptors for external or internal cues. Also, if there is a mutation in the RAS, then it locks the cell in an active state that keeps allowing it to divide. So you can see on the right, normal cell division. And then you can also see if a damaged cell occurs, we result in apoptosis like I mentioned. But in a cancer cell, totally different. If we're in an active state, we're going to divide, even if we're damaged. So this comes to the second point. Cancer cells do not default to apoptosis. These cancer cells have no p53, no DNA damage block, therefore, no opportunity for apoptosis. Even if the DNA is messed up, it will allow these cancer cells to keep dividing and passing on damaged DNA to new cells. Oh man, cancer. Third, cancer cells are considered immortal in the fact that they will never cease dividing. So like I'm saying, they're dividing. They pass through these phases of the cell cycle repeatedly. They pass by these checkpoints. They don't require signals. They're just doing whatever they want. So the next point, number 4, cancer cells are genetically unstable and have chromosome mutations. So think about it. We keep dividing, bypassing the checkpoints of the cell cycle. So if there are any mutation, we aren't fixing them. We are passing them on, and it's going to get worse and worse and worse and worse for every generation of daughter cells that are produced. 5 is cancer cells are extremely invasive in the fact that they lack cadherin and other proteins for cell junction. Cell junctions link cells together through physical contacts, allows them to communicate with one another privately, and allows many mechanical stresses to be shared across multiple cytoskeletons. Without cell junctions, cells are allowed to slip away and metastasize, and it even would affect the private communication between cells. We would just have a whole bunch of public communication, and cells would not like that, because if you're not supposed to get that cellular cue, why are you getting that cellular cue? Well, that's what cancer cells do. Cancer cells, for number the last extreme sociopathic cellular condition of these cancer cells, they adhere to, survive at, and they divide in these foreign tissues of other cells types. So this is scary, because once these cells are metastasized, we are in trouble. We are in massive trouble. The free realm to have these cancer cells in every part of your body, they are escaping, they are going to every part of your body, is hard to control even with treatment. So the last thing I just wanted to touch upon a little bit was that there are different types of cancer cells, and when we are metastasized, what I mean is that we are leaving. So shown on the right, these cancer cells are transported by the circular system to various sites away from where it was before. And they just reinvade and grow in a new location. They can do that. They have no problem going into a tissue where they weren't originally supposed to be. And that's what makes cancer so lethal to people. And it's so hard even with treatment to control. And on the left, I'll briefly mention there's malignant versus benign tumors. Benign are just very localized, and these have a high rate of getting treated. But malignant is a little more difficult. You have to decide between surgery or basically radiotherapy, radioactive therapy. So, I mean, that's kind of difficult. But I think that we are on the rise to come up with new therapy treatments to hopefully help patients and give them a better survival rate, because these tumors and these cancer is just not going away. It's here to stay. Thank you for listening.